Good afternoon. I'd like to thank you all for joining me today and thank Hanson Wade for organizing this conference and for allowing me to speak today on behalf of Hera Biolabs. My name is Fallon Noto and I'm the Director for In Vivo and Oncology Services at Hera Biolabs. For those of you unfamiliar with us, Hera Biolabs is a contract research organization based in Lexington, Kentucky, with a focus on developing cell-based and rodent-based models for preclinical oncology. Today I'm going to focus on our SRG Rat, or the OncoRat, and its utility in preclinical oncology modeling and efficacy testing. I've been with Hera for about 5 years now, leading our efforts to characterize this novel immunodeficient rat strain and its ability to support a variety of xenografts. In addition, I lead our in vivo client services. Over the last 5 years, we've run a large number of client studies, and through these, we found many examples where the SRG Rat has overcome barriers for oncology research. Today, I'll be presenting several non-proprietary examples to you. Hera was spun out of a gene editing company called Transposigen Biopharmaceuticals in 2015. Transposigen's focus was on cell line engineering and custom animal model creation using gene editing tools such as the piggyback transposon system, and together with Hera developed a novel gene editing system called Cas-CLOVER. Hera has the capability and freedom to use these systems for custom animal model development and cell line engineering, which is useful in creating isogenic cell lines when assessing a particular target gene of interest, such as with patient-derived xenografts. The topic of my talk today is on an animal model that was created using our proprietary gene editing technology, the SRG Rat, and specifically on its utility as a valuable addition to the oncology research toolbox, which we've coined the OncoRat. At the end of my talk, I'll briefly touch upon additional uses of the SRG Rat, including creating patient avatars with PDX tissues for personalized medicine, and immune humanization for immuno-oncology studies. The SRG Rat was built on the Sprague-Dawley background. The R refers to RAG2, which is involved in VDJ recombination. The G refers to interleukin-2 receptor gamma chain. These two combined mutations result in a loss of mature B, T, and natural killer cells. The rat is the preferred rodent species for metabolism and toxicology studies, allowing for smooth transition of activities from discovery to development. This slide shows FACS analysis of various tissues in the SRG rat compared to a wild-type Sprague-Dawley rat. Top panels are representative from wild-type Sprague-Dawley, and the bottom panels are representative from an SRG rat. On the left are analyses of the thymus, which is absent in the SRG. Tissue harvested from the presumptive thymic area is devoid of mature T cells, compared to a large population in the wild-type rat. The middle panels show staining for mature B cells marked by double positivity for IgM and B220. You can see that the wild-type rat has a large population of mature B cells in the spleen, which are completely absent in the SRG. The right panel shows natural killer cells present in the wild-type spleen and a smaller population in the SRG rat. We've done some further analysis of these natural killer cells and have actually shown these residual natural killer cells to be non-functional. We've also characterized the immune populations in the blood of the SRG rat and have compared them to the RAG2 interleukin-2 receptor gamma chain double knockout mouse. As you can see here, both the mouse and the rat have similar numbers of B, T, and natural killer cells in the blood, but there are obvious differences in other populations, including myeloid lineages, neutrophils, and macrophages. So while these two animals both have the same two genes knocked out, there are obvious differences in the immunophenotype. Now I'll move on to tell you about the work we've done in the OncoRat with regard to prostate cancer xenografts. Our aim was to determine how well the OncoRat supported several different cancer cell lines, knowing that prostate cancers have historically been difficult to grow in commercially available immunodeficient mouse strains based on discussions with experts in the prostate cancer field. The data I'll show you involve VCAP, enzalutamide-resistant VCAP, LNCAP, and 22RV1. Our first endeavor was with VCAP, a well-known AR-expressing human prostate cancer cell line. Here, we show tumor volumes in the rat versus NSG mice. The first thing you'll notice is that the rat supports significantly larger tumor volumes than the mouse, and that these tumors reach these large volumes sooner compared to the mouse after inoculation. One question we frequently get is on cell numbers. How many cells do we put in the rat, and how does this compare to what is inoculated into the mouse? For these studies, which involved subcutaneous tumors, we inoculated 10 million cells into the rat and 5 million cells into the mouse. And based on the animal's weights, this equates to about 40,000 cells per centimeter square body surface area in the rat and 70,000 cells per centimeter square body surface area in the mouse. So in this case, we're actually putting in more cells into the mouse per body surface area. Our take rates in the rat are over 90% with VCAP, which we've been able to reproduce in multiple growth and efficacy studies. The large size of the rat and larger blood volume allows for serial blood sampling, which is useful for biomarker analysis. Here we show that PSA, prostate-specific antigen, can be measured through weekly blood draws, and this correlates well with tumor volume. In addition to weekly blood draws, the large tumor size supported by the rat allows us to collect fine needle aspirates weekly, which may be beneficial in assessing biomarker expression in response to drug treatment. These fine needle aspirates do not affect tumor growth. Here we demonstrate that AR, androgen receptor, and PSA can be detected in weekly fine needle aspirates from VCAP tumors. We've also performed fine needle aspirates on tumors formed from multiple other cell lines without adverse effect on tumor growth. Finally, we show here that as expected, VCAP tumors express AR and PSA by immunohistochemistry. It's important to note that although the tumors grow rather large in the rat, these do not lead to a non-tumor phenotype, but instead feedback that we've gotten from clients suggests that these tumors actually better represent human tumor architecture. We've performed efficacy studies using VCAP as well as enzalutamide-resistant VCAP tumors, denoted here as MDVR. For these studies, we tested efficacy of enzalutamide and UT34, a next-generation androgen receptor degrader. This molecule showed rapid clearance from mouse liver microsomes, but studies suggested that clearance from the rat was more comparable to that in the human compared to the mouse, and therefore a rat xenograft model was necessary to test efficacy. On the left is a graph showing percent change in tumor volume in parental VCAP tumors after treatment with either vehicle, enzalutamide, or UT34, the latter two demonstrating efficient tumor growth inhibition compared to vehicle control. The right graph depicts efficacy of UT34, but not enzalutamide in the enzalutamide-resistant VCAP model. Both of these studies involved castration-resistant growth. We've also tested the ability of LNCAP and 22RV1 to form tumors in the OncoRat, two other popular prostate cancer cell lines. 22RV1 grows well in the mouse, and we see the same in the rat with 100% take rate. In discussions with collaborators and clients about LNCAP, we've learned that efficacy studies with LNCAP often require inoculation of 1.5 to 2 times the amount of mice needed for treatment. This would not be the case with efficacy studies in the OncoRat, as we see 100% engraftment with LNCAP tumors. Shown here also is that PSA can be measured with weekly blood draws in LNCAP tumor-bearing animals, and that PSA levels correlate with tumor growth. Additional molecular analyses and efficacy studies are ongoing for these two tumor models. In summary, for this part of the talk, I hope the data I've shown help you appreciate the utility of the OncoRat for prostate cancer modeling and efficacy studies. We've demonstrated reproducible high engraftment rates and favorable growth kinetics for 4 prostate cancer cell lines. The OncoRat supports the growth of castration-resistant as well as enzalutamide-resistant prostate cancer. The large size of the rat supports larger tumor volumes and provides more blood volume, and together these features allow for serial blood and tumor sampling, which is advantageous for real-time biomarker assessment throughout the treatment regimen, as well as pharmacokinetic analysis. Furthermore, we've been working with a collaborator at the University of Tennessee who has been able to establish prostate PDX models from primary patient tissue, despite previous unsuccessful efforts in mouse models. Now I'm going to show you an example of how the SRG Rat overcame translational barrier, allowing an efficacy study to be conducted with an ovarian patient-derived tumor. This particular cell line was established from a patient with an actionable gene mutation. The goal was to create an isogenic cell line with and without this mutation. The main problem was that this cell line took over 4 months to form measurable tumors in NSG mice, and the take rate was incredibly low, resulting in the inability to carry out a treatment study in a time and cost-effective manner. In addition, there was a need to verify expression of the target gene after tumor development and prior to treatment initiation, and a strong desire to run the efficacy study in the mouse due to having historical PK and efficacy data of the treatment in other mouse models. So we sought to solve this by first growing the cell line in the OncoRat and serially implanting tumor fragments into the NSG mouse, similar to how PDX tissues are serially implanted. I'll give you the punchline first. It worked. The tumors formed much quicker in the OncoRat after cell inoculation compared to the mouse, and the tumors grew to a large enough size to cryopreserve tissue fragments for multiple efficacy studies. In addition, we performed fine needle biopsy to confirm target gene expression before implanting in the mouse. Finally, we saw faster growth in serially implanted NSG mouse versus single cell injection into the NSG for efficacy study. This graph shows mean growth kinetics of the tumor grown in 3 SRG rats. We had a 100% take rate in these 3 rats. By 100 days post-inoculation, we had ample tissue for fresh serial implant into NSG mice, as well as for cryopreservation for future studies. We could have harvested these tumors much sooner than 100 days post-inoculation, but we wanted to collect as much tissue as possible to expedite future efficacy studies. In addition, we were able to perform fine needle biopsy to confirm target gene expression before implantation into mice. So we sectioned the tumors from these rats into 2 by 2 by 2 millimeter fragments and implanted them subcutaneously into NSG mice, which resulted in growth in 90% of the mice. Tumor development and treatment was initiated within 5 to 6 weeks of implantation in the mouse. Treatment with the test article demonstrated significant efficacy compared to vehicle control, demonstrating the ability to run treatment studies after cross-species tumor growth. While the overall timeline for single-cell inoculation into the rat to treatment in the mouse might not seem like it is much faster than having inoculated single cells into the mouse for tumor development, the point is that the efficacy study would not have been feasible in the mouse without starting with at least twice as many mice as needed for treatment due to low engraftment rates. Furthermore, future efficacy studies can now be carried out in as little as 6 weeks with the cryopreserved tumor fragments from the rat. I hope I've been able to convince you that the OncoRat demonstrates enhanced engraftment and growth kinetics of multiple human cancer cell lines, and this allows for faster model establishment. The OncoRat has been useful in creating banks of xenograft tissue that can be used for downstream efficacy studies, and these efficacy studies can be carried out in either the OncoRat or SCID mice. Finally, the OncoRat can be used to create banks of tissue from either commercially available cell lines or primary patient tissue or PDX tissue that has been established in SCID mice. In the last few minutes here, I'll highlight some of the insight we've gained from PDX studies with non-small cell lung cancer primary patient tissues that we've conducted in-house, and also briefly touch upon some of the promising immune humanization data that has been generated by one of our collaborators. In our own efforts to develop PDX models, we've successfully established non-small cell lung cancer PDX models using the OncoRat. Similar to tumors grown from cell lines, tumors grown from resected patient tumors grow larger in the OncoRat, and in most cases, this growth occurs over a shorter period of time compared to the mouse. This leads to faster timelines to bank establishment. We can harvest sufficient tissue at the first passage from 3 to 4 rats for immunohistochemistry, genetic sequencing, cryopreservation, and serial implantation to expand the bank at the second and third passages. With the OncoRat, there's really no need to go past the third passage. We've also been able to grow OncoRat established PDX from passage 1, 2, and so forth in the NSG mouse. Lastly, for those interested in immuno-oncology, we have a collaborator at the University of Pittsburgh who has humanized the SRG rat using the BLT method. This involves implantation of liver, thymus, and/or spleen tissue from a human into the kidney capsule, simultaneously with CD34 positive hematopoietic stem cell injection. The rat was beneficial here due to its large size and ease of surgical manipulation. After engraftment of the tissues and the cells, the CD34 positive cells develop into mature human lymphocytes and can be detected in the human thymus, express CD3 for T cells, CD68 for macrophages, and can also home to other human tissues implanted. In this case, the researcher also engrafted full-thickness human skin into the rat, which is viable, makes proper connections with the rat skin, and grows human hair. This model is useful in infectious disease. For this particular study, they demonstrated the ability to infect the skin with MRSA. It may also be incredibly useful for immunotherapy applications. One additional difference here between the rat and the mouse is that the rat humanized with this method had no apparent graft versus host disease, whereas the mouse does. I'd like to wrap up by summarizing the takeaways from my talk. While the mouse models have been instrumental in oncology modeling and preclinical testing, we believe that the rat is an invaluable addition to the oncology toolbox based on high engraftment of a variety of human tumors, fast and uniform growth profiles, which are important for efficacy studies, larger tumors, which aid in serial tissue sampling throughout the treatment regimen, and resulting in a large amount of tissue that can be harvested at termination to allow for multiple modes of analyses, and the historical preference to use a rat for pharmacokinetic and toxicity testing due to these parameters being more human-like in the rat. Overall, the rat could save a lot of time, effort, and animals when used in oncology modeling. As I mentioned at the beginning of my talk, Hera Biolabs is a contract research organization. Some of the services that we can provide are efficacy studies in the OncoRat or SCID mice for side-by-side comparison of the two species, PDX establishment or expansion of your favorite tissue bank, pilot tumor growth kinetic studies. We can also provide SRG rats that are bred and shipped from Charles River Laboratories. And finally, we can provide OncoRats pre-implanted with tumor models. We're continually expanding our list of validated xenograft models, and here are a few. We have several non-small cell lung cancer commercially available cell lines established, as well as a collection of PDXs, prostate cancer, colon cancer, ovarian cancer, breast cancer, pancreatic cancer, kidney cancer, glioblastoma, and leukemia and lymphomas. Lastly, I'd like to mention once again that we have a variety of proprietary gene editing technologies and can also provide services with these. These include the Cas-Clover system, which are like molecular scissors useful for introducing knockouts, knock-ins, and base pair edits, and the piggyback transposon-transposase system, which has the capability to introduce small to very large genetic cargo into your cell line or animal model of choice, resulting in footprint-free gene editing. With that, I'd like to thank the entire Hera Biolabs scientific team, pictured here, doing some team building at Keeneland, without whom these studies would not have been possible, especially the entire in vivo team who carried out the animal studies. I'd also like to thank Dr. Narla and his lab at the University of Michigan, Dr. Billady and his lab at the University of Pittsburgh, and Dr. Narayan and his lab at the University of Tennessee. I'd like to thank you all for taking the time to listen today, and also once again thank Hanson Wade for organizing this virtual conference.